summarize my best this topic. When we think about biomarkers, we should go a little bit back and think about what precision medicine is. And precision medicine points the way that biological markers, so-called biomarkers, can be discovered by omics technologies. It means that we can analyze the composition of biological systems, either it be a cell, tissue, or organism, in response to therapeutic agents or diseases. And the development of these technologies has enabled to study the underlying biological processes of ocular surface diseases in more detail. And in the slide we can see that the omics can spread throughout genomics, transcriptomics, proteomics, metabolomics, so all these great words to say that according to the type of the composition, the molecule that we are studying, it has a name, but overall it's the omics technology. In the case of ophthalmology, we will call them oculomics, so a branch of the precision medicine that allows us to study ocular biomarkers and putting the emphasis on identifying clinically accessible signs, biomarkers that can predict disease risk, monitor treatment response, optimize outcomes, and also provide safety, and also understand the eye and systemic health. So this historically could be the first biomarker that was the idea, the concept of biomarker coming with Sir Benz Jones when he pointed out for this immunoglobulin-free light chain that was present in 3/4 of the patients with myeloma. There's a complex classification or taxonomy of biomarkers that can be genetic or molecular biology, their characteristics and all their clinical applications, and we could go through down this tree, which we don't have the time right now, but this is just to give the idea of the complexity of the biomarker realm. So biomarkers are characteristics of biological processes, said before, that can predict relevant clinical outcomes across treatments and populations, and they are independent of the subject's perspective, opposed to clinical endpoints, which are variables that reflect subject's perspective. And surrogate endpoints are biomarkers that can be entrusted to stand in for a clinical endpoint without replacing. So these are three different concepts that we can find in literature. An ideal biomarker should be specifically targeted to improve prediction, diagnosis, prognosis, and monitorization of treatment, as said before, and should hold the potential key for personalized medicine, for the precision medicine, and also by that enhancing clinical outcomes. And we have in the diagram the characteristics should be reliable, safe and easy to measure, sensitive and specific, can be applied to diagnosis and staging prognosis, cost efficient for follow-up, and modifiable with treatment. And with all these characteristics, we can easily imagine how difficult it is, how complex it is to say that a biomarker can fulfill these roles. So why do these matter in terms of the tears? Dry eye disease is driven by a self-perpetuating inflammatory cycle, as we have seen earlier, and biomarkers can provide insight into disease activity, subtype treatment selection, and response. And on the other hand, tear and ocular surface biomarkers can also inform about pathogenesis in early diagnosis, prognosis, and also help managing systemic disease as we started this morning. So healthy ocular surface is a vital component of a well-functioning eye, as we have seen, and this is a complex system involving the cornea, conjunctiva, lids, exocrine glands, and their innervation, forming this integrated entity which regulates the production of tear fluid, which protects the ocular surface against environmental pathogens, accelerates wound healing, ensures sustained eye comfort to provide optimal vision quality. And we can find there a lot of analytes to give us potential biomarkers, as we can see, lipids, nucleic acids, and so on and so forth. We will see them in a minute. And having also this notion that it's secreted and produced by the lacrimal and meibomian glands, and also conjunctival goblet cells into these two distinct layers, as we have seen also earlier. And another concept, and we have just to remark or to underline what we have seen, is this vicious circle that underlines the dry eye disease. Dry eye disease can be categorized in aqueous deficient, evaporative, or mixed. And if in the past, Sjogren's syndrome was the one only entity with dry eye associated with inflammation, as we have seen earlier as well, and now I would like to emphasize, is that inflammation is exactly the central vicious circle, kind of engine for dry eye in every, either it be Sjogren or not. So we have a rich proteome. We have also lipids, mucins, water, ions that we can measure in capture and gather in ultra levels. It's quick, inexpensive, non-invasive, and small sample volume collections. So it's good to have as a source of biomarkers. When we think about what we can extract from the tears, we can start by the proteins, and we can see in this slide a bunch of proteins that we can study, since epidermal growth factors, as we can see, proteins that are blood derived, proteins that can be help us defending against pathogens of the surface, immunoglobulins, as we can see, cytokines, chemokines, etc. Nucleic acids, and it's a word of remark for the microRNA that is crucial for regulating gene expression. It's kind of a guard that can alter what comes from the DNA to the RNA into the cell for the ribosomes to produce proteins, and this guys, so to speak, microRNA can alter all this communication. So it's very important. It's found elevated in all these entities that we can see here, since dry eye to a lot of other diseases that can be systemic, as we can see, for instance, diabetes mellitus, Alzheimer, etc. So the stability, straightforward detection, and accessibility of microRNA make them possible, or makes it a promising worth of work for clinical applications, but it's still in these early stages. Lipids, again, we have covered all this in a previous presentation, beautiful presentation, just to emphasize that meibomian gland dysfunction can be very the key for, or the dry eye that we have in front of us, or at least have a very good role in aggravating the symptoms, and having all these notions, and being able to have some kind of a hint in the tear that can kind of highlight this is important in the clinic. Mucins, we have also seen previously that there are these two types of mucins, glycocalis and also goblet cells. A word of remark for the mucin that is 5AC, which seems very promising in terms of laboratory to give us a good response, and also the importance of glycolization to allow that all these mucins can really protect the ocular surface. Metabolites, a lot of them, taurine, glutamic acid, glutamine, etc. And we can also recognize them in some tears that are used in clinic. The metabolome is nevertheless less studied than the proteotum, and also so it still has a lot of a way to go. And also would like to emphasize the importance of these components that are so-called the extracellular vesicles. So these are lipid bilayer enclosed membrane structures that are very varied and that are shared either from the plasma membrane or also from the endosomal system. So they are formed outwards through the membrane or coming from the inside. And this varied group includes also apoptotic bodies, microvesicles, and exosomes. And the composition of these structures depends of three elements: the cell of origin, the process of their formation, and the functional state of the cell. And what was said is this variability makes exactly the extracellular vesicles ideal carriers of potential disease markers. So these kind of carriers, these vesicles, have a lot of information inside of them that can vary along the process of their origin, formation, and the cell that they are origin from, and provide us important information in terms of what a biomarker can do. So it has been identified in dry eye disease, Sjogren, for instance, thyroid disease, Graves' disease, diabetic retinopathy, etc. The last one, but not the least, are the neuropeptides as players in the axis, the crosstalk in a neuro and immune system. So what happens is, by means of a pathogen or mechanical damage, what happens is that the sensory nerve endings which populate the cornea, as we have seen earlier, become damaged themselves or stimulated. And they start secreting peptides, so small protein molecules. And for instance, we have here identified the substance P, calcitonin gene related peptide, vasoactive intestinal peptide, and the neuropeptide Y are also examples of the products that these sensory nerve endings can produce, and can by themselves trigger and regulate the neurogenic inflammation response that can have implications on the wound healing, protection from infection, and also elucidate us about the pathogenic mechanisms, for instance, in dry eye, and all these diseases that are depicted in the slide. And also a word of remark again for systemic diseases as well. And having the notion of the axis between the central nervous system and the eye, and how this can be taken into our advantage as ophthalmologists, and also collaborate with our colleagues of the other specialties. So these are the ones, the semi-quantitative markers that are more common, as you can see. We're not going through them. And then we have this great table, and we have a lot of names here, and the names of these molecules with these sometimes strange names, they usually can help us in terms of inflammatory mediators, tear film stability or instability, stress of the epithelial, immune activation and chronicity, neural feedback alterations, and also differentiate Sjogren syndrome versus other entities of dry eye disease. And this is another one focusing the key biomarkers of dry eye disease. And this is impossible to describe. It's with the time that we have. And also, it's just to emphasize the complexity and the pathway that is so exciting of investigation, working hand by hand with ophthalmologists and the fundamental investigators and other specialties, and how rich the horizon can be in the future. So why doesn't this, or why is it so complex and also difficult? The variability in tear fluid analysis. There's no standardized method for tear collection, analysis, interpretation, and reporting. And so the small samples that we can find in the reports and scientific studies, these are the three main reasons that explain the low reliability and reproducibility that we still have in what we say and the difficulty in summarizing all this information, how we can interpret really and accurately the data, and also the delay in translational science. So the clinical use was this one. I would just like to show the last slide to make it, if please, just the last one, which is this. And the summary is that dry eye is the self-perpetuating inflammatory cycle. So if we have some biomarkers, and these are the most validated that we have there, the metalloproteinase 9 and also tear osmolarity with some limitations. Strong candidates, we have them there as well. And the future direction will be for sure to look at those clusters of biomarkers and think how they can help us in signaling a disease, instead of going one by one, but having them as a panel, making them work with each other so they can inform us about diseases of the eye, prognosis, predictions in terms of what is going to happen, responses to treatment, but also related with systemic diseases, Parkinson's disease, multiple sclerosis, for instance, just to give an example. Thank you so much for the time.